All right, this video is going to go over protein anabolic pathways, more specifically on how do we make proteins within the body. This orange box is here. So essentially, we can use protein that's broken down from our body using those amino acids in this amino acid pool. And we can use dietary protein that's been broken down into amino acids and contributed into this amino acid pool. From this amino acid pool, we will then be making those proteins within the body, such as structural proteins like your skeletal muscle, nucleotides for DNA, apoproteins for lipoproteins, enzymes such as lipase, and then hormones along the lines of glucagon and insulin, to give some examples. So the process in making proteins is step number 1, cell signaling. Step number 2, transcription. Step number 3 is translation, and then ultimately creating that protein structure. So this is kind of an overview. When we're looking at the individual cell within the nucleus, we are going to split apart that DNA, and we're going to basically code and transcribe to get an mRNA strand, which then goes through this cycle of the ribosomes inside the cell to be translated. And based off of this unique genetic code, it is going to then stimulate an amino acid chain to then make your protein. So that's a really fast overview. We're going to go through each of the steps. So step number 1 is cell signaling. This is going to initiate protein synthesis within the cell, again, triggering the nucleus and then the ribosome to create the protein. This is highly regulated, and its different cells require different proteins. So the first step in this cell signaling is going to be separating the strand, creating a new strand, as well as proofreading that strand to ensure that there's no DNA mutations. So again, as I mentioned, this is a highly regulated, so we're going to actually reduce the amount of proteins that we make, or we're going to increase the amount of proteins that we make. For example, if you are newly engaging in resistance exercise and you need to make more skeletal muscle to support your muscular growth, you're going to uptake that amino acid more. Whereas if you decide to all of a sudden follow a vegan diet and you no longer need lactose, you're going to downregulate or reduce the amount of lactase that you are creating inside of your body. When we are looking at the DNA and RNA replication, again, we are looking at the nucleus and we are splitting apart this DNA strand that has all the nucleotides, which we've looked at how they match earlier in class. And we're creating a complementary pair to make an RNA strand. Now, this does require initially an RNA primer, which is going to be at least a few pieces of this nucleotide. And then the DNA polymerase 3 is the enzyme that's responsible for actually synthesizing the DNA strand, and it does require nucleotides. And then we also do this proofreading, which is the 3', 5' exonuclease. And this is going to basically be your spell check. So it's going to catch about 99% of those mutations if for whatever reason a complementary strand does not get attached. Again, this is a reminder here that thymine is DNA only and uracil is RNA only. Now, when we're looking at transcribing, this is the basically blueprint. And so we are reading this DNA strand to decide what we actually need to make in that RNA strand. And so then we are able to basically code and create or translate and form a polypeptide strand. So if you think about it, transcribing is kind of the raw data, and then translation is the information that gets translated from that raw data to make the specific protein. Again, a reminder that protein strands are unique in regards to each of these amino acids and the sequencing of them, and that's what determines what type of protein is actually being created, whether it is an enzyme or a hormone, or whatever type of apoprotein that might be needed inside the body. This is just another picture that can show you, this is our DNA strand, and we are going to transcribe that into basically a blueprint mRNA. And then we're going to actually build the building using that blueprint. So that's when we're translating and using the individual amino acids to form the polypeptide chain. From there, we're going to use the final stage to create the protein structure. So based off of the type of protein, we're either going to have primary, which is just the linear chain of amino acids, the polypeptide chain. We could have a secondary protein structure. We could have a tertiary protein structure, or we could have a quaternary protein structure. So this would be the completion of protein synthesis, is creating the actual structure that's needed for the individual protein.